I'd like to do today is address what I think has been one of the great successes of this meeting over the years, which is the challenges to clinical translation and preclinical translation of research. So I'm actually just going to talk very briefly about my own work, just in 4 slides, and just quickly run over the 4 clinical programs that I've been working with, and then spend the bulk of the time really talking about the challenges that I see in translation in the preclinical setting, and then translation that I've learned about in the clinical setting. So to substantiate those views, I just wanted to let you know that I have had some experience doing some translating. With Professor Tom Lane, we developed the proof of concept, and then I commercialized the CXCL10 antibody for immunotherapies. We showed this molecule worked in multiple sclerosis to attenuate multiple sclerosis, and then I translated that into spinal cord injury and showed that it was an effective way of reducing secondary degeneration after spinal cord injury. And we tried to get it out there in the world, and we couldn't, and we learned something, that the spinal cord injury field is just too small to translate a drug into for big pharma. So we then translated into ulcerative colitis and rheumatoid arthritis in preclinical indications, broadening the market out to a cumulative $6 billion. Then we had people pay attention to us, formed a company, the company was taken over, and the program is now at Bristol Myers Squibb. So this program I was involved with in inventing it, doing the preclinical, and right up to the clinical approvals of phase 1 before it was handed off. What's happened since is 2 clinical trials of phase 1 and a multinational phase 2, actually 2 phase 2s, multinational, and the latest trial, over 87, I believe, % efficacy in ulcerative colitis, so very, very happy about that. But I learned how to move something along, and that was my first foray. Then I worked with Geron Corporation, as Ed told you about, and developed a cell population with my team at the University of California, Irvine, of making embryonic stem cells into oligodendrocytes. And it was really the first high purity source of cells made from stem cells, and therefore it became the first clinical trial. And it has gone pretty well, besides the company halting it for financial reasons. But as Ed told you about, 5 patients treated, and safety was demonstrated in an interim report for those first patients. I then, with California Stem Cell, developed another technology, taking embryonic stem cells and making them into high-purity motor neurons. I originally conceived of this as a treatment for spinal cord injury, but again, because of the small market size and my experience with clinical translation, I decided to move it first into spinal cord diseases, and in fact a terminal pediatric disease, which is a fairly accessible clinical entrance. So this population is going to be tested first in babies with spinal muscular atrophy. They manifest at about 1 to 3 months, type 1 SMA, and then 95% of them die by 9 months of age. And just a horrible disease whereby the motor neurons are lost low in the spinal cord, and they have flaccid paralysis of their legs, and then moves up, and you have digestion problems. Eventually they can't suck or digest, and they starve to death or die of respiratory failure if they're intubated. The babies live such a short period of time, it's a very quick clinical translation, because they all die within 6 months of manifesting disease, or most of them do. You get through your phase 1 and phase 2 clinical trials quickly, and the sponsor can enjoy valuation jumps. Sounds sort of callous, but it's an indication of how monetization of these things is required, especially in orphan diseases. So it's going to enter babies with SMA first, and the phase 1 clinical trial was proposed to the US FDA, and they came back and they requested a second safety study, preclinical safety study, even though we did the precise safety study that they asked and we negotiated with them. And then when we revealed that safety study, which was a resounding success, they said, please do another one in a different rodent model. We did it in a non-skid mouse. They wanted a nude rat. in addition. So we took it over to England to the MHRA, and you should all know that stem cell policies have largely been originated in the United Kingdom. The US has adopted largely the policies of the UK. So very respected policymakers, very respected regulatory body, the MHRA, and they actually said that no such trial need be done because it was likely litigious in nature that the FDA required this, fear of litigation. And whether they're right or not, I don't know, but it is an indicator that that US is much more cautious than other high-standing regulatory bodies. So we will submit the final application, I believe, in January, but one of those two routes will be opened up quite soon. So this is just about to be translated into babies with SMA, and then we'll follow it up 3 months later with ALS and then spinal cord injury. And then lastly, I've recently been working with my team at California Stem Cell to develop and refine a manufacturing regime for cancer. And this I'm really, really excited about. We've not really put this out there yet in the scientific and medical literature. In this therapy, what we do is we take the tumors out of the patient, and with late stage melanoma, stage 4, we pull the cancer stem cells out of the tumors, and that's the uniqueness. It's the only approach doing that. So the cancer stem cell was identified a handful of years ago, about 5 years ago, and it's now known to be the root cause of cancer, if you will, in that they drop several seeds, and those seeds grow into tumors, and 5 to 20% on average of a tumor is actually cancer stem cells in the middle of the tumor dividing and making the bulk. You can have cancer stem cells fall asleep in your body for years. So if you undergo radiation or chemotherapy or surgical resection, those sleeping cancer stem cells are alive and they're missed, and you still have cancer. That's why chemotherapy and radiation and surgical resection rarely work. So what we've done is by taking the cancer stem cells out of the patient's tumor, an autologous, and then taking their dendritic T cells, dual autologous, educating your own immune system against your own cancer stem cells, reintroducing the primed dendritic cells back into your blood system, and it's very simple. It's just needle injections at the clinical site. It's quite a wonderful thing for spreading around the world because we're not asking much of clinical participants. We've got, in our last clinical trial, we just ended it, and we have a 72% survival rate at 2 years, which is on record the best cancer result in any phase 2 to date. So I'm very, very excited about this. We've worked in refining the manufacturing protocol so we can pull the cancer stem cells out of every single tumor, and we can do it all within 6 weeks. And we've thus far completed the phase 2 clinical trial, 2 of them actually, 2 clinical trials on melanoma. And we're just starting a clinical trial on liver cancer, and we've shown at the bench that we can pull the cancer stem cells out of ovarian cancers. So it's any bulk metastatic tumor. And we've got a December 14th date with the US FDA to review our phase 3 application. So I'm really excited about this. But it's been a manufacturing issue of how to get this product out there in a commercially viable ramp-up. And I forgot to start the conversation off by saying, by way of disclosure, I'm associated with California Stem Cell and receive some monetary benefit, as well as Johnson & Johnson. So, being involved in those 4 clinical trials, or clinical programs, I should say, what have we learned? Like, what's the landscape of clinical trials? So a couple reality checks and then lessons. So reality check here. Clinical trials have a very high failure rate. So if you look at the likelihood of transitioning from a phase 1 to a phase 2 clinical trial, it's not bad. Chance of transitioning from a 2 to 3 is very poor. And then of course you've taken the safety and efficacy risk slightly out, so your chance of transitioning to market and post-marketing analysis gets better and better. But you have a fairly high failure rate. If you look on average, 82% of phase 2s fail. 50% of phase 3s fail. Just a phenomenal failure rate. It's very, very high. I used to think how wonderful it was when that first one, the IP10 or CXCL10 treatment got to market. So I was at the FDA, looked like we were going to get our approval, and I thought, wow, what a victory. And then I started talking to people like Wise Young and other surgeons, and I realized that's actually just the beginning. It's the start gates rather than any kind of victory. And here's why. It's a hard race after you get into the clinic. Now, they fail for all kinds of reasons, and they fail by application. So central nervous system approval rates are actually exceedingly low when you look at other groups like anti-infectives. So central nervous system in particular is very, very low approval rates. You're talking 92% failures in central nervous system clinical trials overall. So why do they fail? A few categories or a few modes. First of all, technology failures. It's one of the primary causes, of course, or for certain the primary cause is the technology itself is less than adequate. So with regards to efficacy, overall you have greater than 50% efficacy-related failures. So that's certainly the primary reason why things fail. The drug, the cell type is simply just not going to do it. Cell therapy rates in particular should be better. And this is something that is generally poorly recognized. There is significant preclinical data developed in academic settings that is not developed in many small molecule settings. We could talk for an hour about it, but in the cardio field, there's often no preclinical work, zero. It's initiated at drug companies, and it's based on historical literature data, and they go right from a new chemical entity into clinical trials without preclinical work, or very little of it. But with new spinal cord and cell therapy, which is a new emerging technology, there's significant academics that play into it. So you have a tremendous amount of preclinical data. It is safer, and the statistics and analyses do bear that out. And patient data often exists because groups take these things outside of the United States. Like I know the Geron work, while I was doing that initially, it popped up in 4 different countries, in Russia and China. And were they growing oligodendrocytes? No, they couldn't quite do it right. So they called them other cell types, but you get these copycats outside. Nonetheless, it does provide you with evidence that you can use. So the experts tell us that we should expect large molecule approval rates. Large molecules also have significant preclinical work, and their success rates overall, all 3 phases, are about 34%, so a little higher approval rates. With regards to safety, about 30 to 40% of safety-related failures are the cause of the clinical failures. And again, cell therapies do not present the same risk profile because you have limited risk of systemic toxicity, which is one of the primary causes of safety failure. However, you do run risks of graft versus host disease, tumorigenicity, and arrhythmias. Those have been the 3 primary reasons for technological failures in cell therapy modes. Quite unique to cell therapies. Not totally unique, but characteristic of the safety failures. Another major category of why clinical trials fail is the clinical trial design. Poorly chosen endpoints, whether they be primary or secondary. So as an example, disease progression versus overall survival. Disease progression is often very difficult. Overall survival is pretty easy. It can be multifactorial. That results in a lengthening of survival. So you kind of have a catch-all. So our latest cancer trial, our phase 2 was disease progression and survival. Our phase 3 is survival only because it's easier. It doesn't matter what causes the survival. If we go in and choose an endpoint as a mechanism of survival, we could lose if we choose the wrong endpoint. This is more broad. It's difficult to measure clinical benefit objectively, and Dr. Worth showed us very well how that is. Small benefits in spinal cord injury, and Keith also said the same thing, may not be detected with these large endpoint measures. Inappropriate patient population. We just heard about this. A broad patient base versus a very targeted patient base. So with respect to spinal cord injury, how similar are spinal cord injuries? And what risk does that produce? I would say a great deal. It makes for very, very tight inclusion criteria that has other downstream difficulties that I'll talk about. The discontinuity between research and chemical or commercial processes. You lose the magic when the manufacturing process is optimized for commercial production. So it's one thing when we were making oligodendrocytes in my lab by the thimbleful, and it was another thing entirely when the cells were taken over by Geron Corporation and ramped up. I can tell you the product changed. I can't tell you how, but I do know how, but the product definitely changed. So any time that there's a ramp up, there's a tremendous risk of throwing all of your preclinical work out the door. And then control group issues, failure to anticipate benefit in the clinical setting of a control group. This has been the dearth of many, many clinical trials. Another major category is clinical trials management. Lack of clinical operations experience in the spinal cord injury field is particularly prey to this because most cell therapy, being an emerging technology, most of these trials are conducted by startups or academics with little or no experience. And that has been a major problem. These are strategic errors. The technology may be incredible, but they're strategic errors because you're just a small operation and you need to go fast because you need to get some news on the marketplace. You need to go fast because you got another investment round that's going to open or close on you. You need to go fast because you're just running out of money. You can't afford pharma or contract research organization involvement, which is tens of thousands, sometimes $100,000 a month retainers to keep these people that really know how to do it. We are very prey to that in the spinal cord injury field. Wise, if I asked you if you did have an extra $100,000 a month to contract a CRO, then I'm sure the answer would be yes, I would love to have that in setting up the China op. Patient enrollment. Inappropriate inclusion and exclusion criteria, a major killer. Inadequate supply of patients. In the United States, this is a staggering figure. This is a new figure, 2011. 80% of clinical trials in this nation fail to meet their enrollment goals and are therefore halted. 80% of clinical trials. That's phenomenal. It's because there are so many clinical trials going, and as I'll show you, the amount of clinical trials being approved by the United States is going down tremendously. So there are larger and larger patient populations for fewer trials. It's highly competitive. So 80%, that is just a staggering figure. Overall, it results in about 23% of failures across the board. A quarter, 1 out of 4, complete failures. 80% are halted for a time. Data management, poor data capture or entry. Again, something that is the spinal cord injury field is particularly prey to because they don't have these experts helping them. And then bias, of course. Another thing that we are prey to is the spinal cord field because it's often the investigators leading their own studies, or at least starting them and leading them for a particular time period. Another key failure mode is funding. So it's a very challenging funding environment out there. The venture capital field is completely on its head right now, and people just don't want to invest. When they do, they're demanding very onerous terms, and it's very, very difficult for companies to obtain this type of funding. There's very, very limited capital available. Many of the new startups are completely angel funded, meaning high net worth individuals funding the things, like the China Net, like California Stem Cell, 100% angel funded thus far. Millions and millions of dollars, not easy. Inadequate resources, therefore, to correct for errors or redesign trials. And when you see that the vast majority, 82% overall, fail, but only a portion of that is due to the bad drug, that means half the failures roughly can be fixed. It's not the drug. It can be fixed. It's the wrong needle, it's the wrong timing, it's the wrong dose. You need to go back. You always learn something in a clinical trial, but the spinal cord injury field is particularly prey to this because we scrape together resources for this meeting, let alone resources for preclinical work, let alone the millions for a clinical trial, and then we learn something, have to repeat a phase 1 or phase 2, and it's just difficult to do it again. Here is just a few hard facts in that regard. As we move through the clinical road, you can see that the early proof of concept preclinical work it's relatively small money, and that's usually grants. And then you start to get into your therapeutic candidates, and the phase 1 is generally 10 to 15 million. Wow, looks like it's hurting. And it's a lot of money. I'm faced with this right now for this cancer trial. I'm out there in January raising $50 million. Very, very difficult to do. There are venture investments or partnerships. And when you consider that the average time to market is 10 to 15 years, the average cost of a successful drug with the cost of money built into it, for each drug that works, there are several that fail, the total cost is $1.3 billion. Failure rates at just over 90%. Less than 30% of approved drugs recoup their development costs. Less than 30%, that's pretty amazing. So this is depressing you, right? It's a reality check in that it's one thing to have enthusiasm about your running rat with spinal cord injury. It's another thing to have a little bit of forward-looking vision and say, wow, do we have some serious things to get over to make this thing attractive? But there are some rosy outcomes of all of this, of course, primarily marrying it to another indication for spinal cord injury. There's another key failure mode is regulatory. Great many fail. In the pie charts I included earlier, it said the precise amount, so you can go back. The regulatory framework is still evolving. I remember with Geron, it being the first embryonic stem cell based trial, basically the white papers were being generated as we were there. So what did that mean for Geron Corporation? It meant that instead of going to the FDA in 30 days getting a letter back, we went to the FDA and then 30 days later we got a letter back that said, well, we need to convene a meeting, we need to have a position paper, we need to have some focus groups, and there was this back and forth. So what does that mean? That means burn rate. If Geron was burning $3 million a month, then that CEO is sitting there saying, okay, so it's going to take 18 months? Whoa, that's a lot of money. Just to keep the doors open and the lights on, the burn rate of the company, let alone the cost of executing on those things that the FDA demands. Regulatory framework right now in stem cells in this nation are virtually killing the industry because of lack of education. Now they're getting a lot better, and I would say one of the greatest contributions of the Geron story was to white paper the embryonic stem cell field, and in many ways the stem cell field is one of the greatest contributions because it's been faster for the subsequent plays. Some key parameters are unclear, like data necessary to establish safety. That was one of the largest back and forths. I told you of the motor neuron trial. I don't think I'm allowed to tell you about the Geron trial, but it was similar. And back and forth. We did an entire study, presented it, the FDA changed their mind. We had to do it all over again. Regulatory agencies are climbing a learning curve, and they fear bad press, and in this nation, litigation. And I gave you an example earlier of that. Just by way of an example of this, the FDA clinical trials involving cell therapies are increasing. Like, look at that. Phenomenal. Cell therapies. And then if you hone in on stem cells, look at that. Stem cell, 200 applications in 2010. They're getting there. There's a lot of activity, but look at the approval rates of the FDA. Phenomenal. Why is that? I just met with a congresswoman and senator to discuss this, and we're going to meet with the US FDA and just to try to decide to help out the stem cell field a little bit and try to find ways to provide the FDA with more money. And my understanding of it now, and this is uneducated, is that the US FDA just does not have the money to staff people. So I told you about 4 clinical programs that I've been involved with. So I've been back to the US FDA, and I like to go, right? I don't have to go at all times, but it's pretty fun to go there. And the people working on the other side of the desk change every single time. And it's funny that the person that was in charge when I was there with Jaron actually worked for me when I went back with my next one, because she left and was a consultant, because they don't pay very well. And if they don't pay very well, do smart guys like Ed Worth go and get a job at the FDA after going to industry? No. You know, it's difficult unless he's financially sound enough to do it. But what kind of people they attract there, it's difficult. You know, you're dealing with a bunch of people that are very, very good, but it's generally temporary because the pay is not that great. So, just switching over to the preclinical work, so that this is, or clinical translation, and just a few minutes on that. These are what I see as the very major challenges to those of us doing preclinical work, trying to get to our phase 1. One thing at a time. Repeating your experimental findings. There are 2 general classes of repetition, and this is obviously the best. If you truly repeat the work, it's wonderful because we just feel good about it. The risk and efficacy is assessed and reduced, but it's very difficult, primarily due to lack of PI motivation. Why do I want to go repeat, Dr. Horner's work? He did a great job of it. Why do I want to repeat it? He already got the great publication and the grants, and he rocked it. But now I have to repeat it in my laboratory. It's not very exciting for me. I'd rather read about it and watch him do his next thing. With literature support, it's very robust because we can all feel good about it. Intellectually, everybody's doing it. Wow, this must feel great. And you just gave us a few examples of that with a long laundry list of feeling good about using that magnesium. It's ethical with respect to animal use. You don't use a lot of animals. However, you have higher risk of efficacy. Safety studies. You've got injection paradigm safety. How are you going to deliver it? What's the survival like in the way that you deliver it? The injection site safety. You have to look at the site itself. Are you causing necrosis? And is your needle too big and causing damage? Is it sharp enough so you're not causing a contusion injury? Is it handheld and vibrating if it's a baby? Or does it have to be fixed and you can get away with that? What immunosuppression and what the toxicity profile, very important for babies. And then biodistribution, tumor genesis, toxicology, and pain, which are very typical parameters and the major guidelines or pillars that have to be attended to in a preclinical safety study for stem cell work. Just an example, I just did one of these. We had 8 months of design with the regulatory QA officers and the FDA itself. 8 months to design the study, an extremely specialized procedure, hundreds of animals, and then we outsourced it by necessity to a third-party GLP laboratory full of a bunch of researchers and technicians without a bachelor's degree. It's high school kids that just got jobs and they're great animal handlers, but they are doing the work now that my specialized postdocs, PhDs, and I do. And boy, that's risky. And then the FDA changed their mind in order to repeat. They just changed their mind. We did precisely what was requested, but they just came up with other stuff. Now, I'm not saying that it was wrong, but it just happened. And it was extremely expensive. This one was about $2 million. So we had to do another $2 million study. And the burn rate. Large animal studies are another challenge. One has to ask oneself, what's the validity of existing models? Is there really a good contusion model out there in monkeys? And has it been sufficiently characterized? You must ask yourself those questions. The cost is phenomenally expensive, thousands of dollars as opposed to $20, tens of thousands of dollars per animal sometime. The skill set that's required is phenomenal. There's ethical considerations, and the necessity, one must note, is dictated by the biology of the cells. It's not necessary to go to larger animals for many cell types that don't have size issues. If you're growing an axon a long distance, you should probably go to a large animal. If you're wrapping fat around a little wire, like oligodendrocytes will wrap fat around a bloody glass tube. And there's no size issue there. And it's already known that they myelinate every single animal. So is it really required for efficacy? Probably not. Safety, maybe. But depends on the biology of the cells. Instrumentation. Medical devices currently in the spinal cord injury field, and Ed Worth deserves a medal for this. They're the focus of individual companies, but no disrespect, but what the heck does Geron know about doing a device, right? You guys did a great job. It was amazing. But instrumentation is the stuff of instrumentation companies. They have incredible tools and engineers to do this stuff, but we're a new emerging field, so we just have to do it ourselves. And that is a risk. Unique skill sets are required for that design and implementation, and it's very expensive as a result because it's not turnkey operation. For example, we had to figure out because we were growing neurospheres or microspheres of motor neurons, over time they grew, but the transplant needle had 160 micron internal bore, so that dictated our instrumentation. There's a lot of work with this type of thing you must pay attention to in the preclinical setting because it may limit your clinical application. 5 out of 10, manufacturing and qualification of cells. Do I have a few more minutes? Oh, yeah, okay. I'm not going to go through all this, but good manufacturing practice just blew me away at how complicated it is. So when I learned how to do this, and I have built with my teams 4 CGMP laboratories now, very extensive labs, 4 different builds, and they are millions of dollars, and they take dozens of very, very intense career dedicated individuals, very specialized, very expensive, and then you run into this. It becomes clinical grade. It was research grade. Now it's a different product. So what is the value of your preclinical work? Because when Hans grew oligodendrocytes in a tissue culture hood in Hans's lab, that's a different product than Geron's once they ramped it up. So it all had to be repeated again. So it behooves you as a researcher to build a CGMP laboratory sooner rather than later so you can lock down and finalize your product. But how can you afford to do that without first proving efficacy? It's a waste of money. There's a lot of wastage right now in the spinal cord field because we're all doing research grade grows, and rather than consolidating and doing CGMP facilities so that we can all use, and then when I come up with a good idea, I just go to this core facility and get CGMP, and then when I spend my $1 million precious dollars and do my study, it's actually relevant to the US FDA. So I am preaching for that. Manufacturing and qualification of the cells. You have GTP, tissue practice, or MP, manufacturing. Scale up. You have quality checks and product checks and process checks. You have batch release criteria. What are you going to say are the right batch release, and what's your specs and what's your actuals? Repetition of primary findings with the final formulation, like I just said. Fairly complex bit of work. The manufacturing and qualification of the media. Don't forget about the media. If you've got a cell product, you must deliver it in a liquid. It is as important in the clinical setting as the cells themselves are because it introduces a risk. Media production, who am I to start making a media? Like instrumentation, media production is the focus of companies that are specialized, but there are none. There's just a few now in the stem cell field that have popped up, but no standardization. Unique skill sets are required for design and implementation. You have to be fully defined and free of rodent reagents, and you have all those same things that apply to the cells. If it's not the CGMP produced liquid, you are working with a brand new product. Finally, the clinical synopsis, or not finally, but almost finally. It's the result of multiple focus groups. It should precede the experimental design, because you really should have medical doctors telling you, don't use that needle before you spend your precious $1 million on your preclinical study, because you may just be throwing that all away because it's of no use, because you really can't do that in a human anyways. So talk to people first. The clinical should reflect the preclinical. That's why this is so important. And you have to select appropriate patients from a diverse range of injuries. So, just in summary here, here's what you generally run through. You gather together a dozen or so medical doctors, which is really, really hard to do. And you discuss which patient population. And then what are the inclusion criteria? And then what's the device? And then how are you going to provide the product? What immunosuppression? With all those things, what's the risk to the patient? And then what are we going to do when they die, or if they die? And what are the outcome measures and the pre and post-op care? What's the clinical continuity? Is there going to be a nurse that guides them through the whole thing? You know, in spinal cord injury, there is no clinical continuity in this country. I think Shepherd set up the first, I think. I might be wrong, Keith, but it's clinical continuity of a patient coming in and going straight through, right all the way to rehabilitation. Just doesn't exist, or just newly exists in this country. Rehabilitation, what's going to be required? Put a bunch of living cells into a spinal cord and you expect them to do something without movement? That's foolhardy. You need to pair a rehab regime. Now, holy cow. Now I have to become a rehab expert. Ethics and site selection. The outcome measures, you know, the benefits are going to be incremental, so we talked about that a little bit. How are these typical outcome measures going to pick up a very incremental benefit? We need graded outcome measures of fine function, which really don't exist for many regions of the spinal cord, like the thoracic regions. Rehabilitation regimes have to be standardized, not only for spinal cord injury, but for types of spinal cord injury. And patients must have this continuity of care to rehabilitation, to outcome analyses from the surgical point, so that they can actually be, the rehab and outcome can be customized to them. And most trials require one primary outcome. It's very difficult to pick these things in a clinical setting. Financing the program. Small spinal cord market discourages investment by industry. I think I covered that. If you walk the path, it's extraordinarily difficult when you see the small market size. The risk is high. Patient recruitment is difficult. Outcome measures are tough. So you may not be able to detect it. You may not be able to get the patients to do it. It's amazing that Geron had to have so many sites, isn't it, for so few patients? The inclusion criteria were strict. Barrier to the IND is high, given the long life of spinal cord injured individuals. I was actually at the FDA once, and outside, talking to one of them, offline, I was told that, you know, it's not really necessary because they live normal lives anyways. And, you know, tell that to a spinal cord injured patient. So all experiments must be repeated with the CGMP material. So how do you get over financing the program? With this company I was involved with, we started in spinal cord injury, and then we expanded it into multiple indications. With the motor neuron treatment, we expanded it into multiple indications. With the cancer treatment, we expanded it into multiple cancers. So that's what you have to do, especially for this area, spinal cord injury. And then lastly, I'd say one of the great preclinical challenges is the relative immaturity of the spinal cord injury field. You know, we're all isolated, we're duplicating each other's results, we don't talk to doctors, we don't have somebody down the hall, industry isn't really involved, so we don't have a lot of industry interaction. And the government, FDA, is still making up its mind on a lot of things. So whose job is it to do all this stuff and consolidate it? When you look at the history of other diseases that have been tackled, it usually emerges from the formation of one very large company that starts to consolidate. They say, Dr. Kosas, you do the monkey stuff over there, and Keirstead, you do that rat stuff, and you do this and you do that, and you know, Wise, go set up trials, if you will, in China, and you have a consolidator. But we're a young field, we don't have that yet. So just two last slides, a couple case studies. So Geron Corporation ran into economic difficulties. And in summary, why? Very high regulatory bar and a slow approval process, very strict patient inclusion criteria, slow patient enrollment. Was it 18 months for 5 patients? Is that right? Reimbursement challenges. They had to walk that path. And how are you going to get reimbursed from this from insurance companies? Long patient follow-ups, 15 years. Phenomenal. That all just means money. Early technologies and extreme scrutiny of a new field resulted in a very high cost of development, and that says small market. Now, what happened? Just as an example, Geron spent about $200 million on its stem cell programs, and it was a decade in development to get something preclinical. $200 million in 10 years. I've done several small company startups, and if I went and sat in front of a room of investors and said, could you give me some money because it'll take me $200 million in 10 years before I get an IND? they would do that. They would laugh. And they won't do it. So it's a different model. Another case study, Stem Cells Inc. Same thing, right? Stem Cells Inc. just started a spinal cord trial, and they're facing the same thing. High cost of development, very small market. How are they getting over this? Well, CIRM right now just gave them a great big dollop of money, very creative. But what's their reality? They were at $171 a share, and today they are at $1.87. They were 8 cents 3 months ago. $1.87. So what's happened? How do you recover from that economically? It's a share buyback. Somebody has to come in and buy up all the shares from the public. But who's going to do that from 171 to $1.87? So it's 16 years in development. I don't know how much they've spent in total, but this says a lot. And it's just got into spinal cord injury. Just right now. So that's my last slide. I don't mean to just depress everybody, but it is the lessons are simply that we have to overcome these things. And one way to do it is for the early researchers who are doing preclinical work to understand what they're getting into so that you can actually look into manufacturing and clinical paradigms prior, so you don't go repeating years and wasting millions of dollars by doing it incorrectly. Knowing where you're going so that you can do it more efficiently can save millions and millions of dollars. So I told you Geron was $200 million in its preclinical. I believe it was $45 million on our program preclinical. California Stem Cells was $5 million. 205, because we already walked the path once. So the second time was cheaper because we knew what we were doing. So you can do it. You just got to know your beast. Thank you.
Thanks, Hans. And so we have a couple questions, but we also have Dr. Kwan waiting on the horn. So go shoot.
Well, I was just going to ask, in light of all this experience that people are having, if you had solutions to this that might involve a completely different approach that isn't, or if it would be possible to have a different approach that isn't industry-based product development for clinical application, but instead, research funding for human neuroscience. Neurosurgeons do things all the time that nobody asks permission for.
Yeah,
the IRB doesn't look at.
Investigator-initiated trials.
Yeah,
absolutely.
and there's a different culture and a different history and a different bar for doing that stuff. And I mean, it used to be you could enroll someone in a drug trial and sort of 1 out of 10 people would meet the inclusion and exclusion criteria. Over 250 people were screened before they got 5 for Geron. And I don't know how much of that was because it was stem cells or because they had all these safety implications or the time window, or whatever it was. But I mean, if that's the view of the future, I'd rather try to figure out a way that it wasn't part of some formal clinical trial, but a way to have 15 different investigators study one patient,
right?
At their institution as part of a research grant. But I don't know if we can get to that.
I think that's a great way to do it. You're basically spreading around the cost. Now you do introduce more risk in there. There's a whole paper I could show you on how risk goes up with the more clinical sites you add. Right? It makes a lot of intuitive sense. So what you're suggesting could work to keep the cost down, but it does introduce more risk and inter-rater reliability or variability, I mean to say. So could be done, yeah.
If I can just make one comment about the Geron program, because I think it's an important point to note, and that is that of that $200 million, only a small fraction was actually directed specifically at spinal cord injury.
Right.
There was a significant amount over several years, just first qualifying the embryonic stem cell banks with FDA in order to get permission to use that as starting material. Then on top of that, there were several key programs in addition to spinal cord injury. In particular, was the cardiomyocyte program and for heart failure, and the islet program for diabetes. Those were really the market drivers all along because they were potential big market. The downside to that strategy is you had distinctly different products for each of those indications. So that was very different than the strategy you've adopted at California Stem Cell, where you're saying, I'm going to focus on a much smaller number of products for multiple indications, as opposed to different products for single indications, which is much more expensive. So anyway, there were some important lessons learned from that I think sometimes get lost in sort of the broad strokes.
Couldn't agree more. And that statistic keeps popping up in my head of that only 30% of clinical costs are ever recouped.
Right.
So
Exactly.
people march the clinical route for various reasons.
Yeah.
It can be the surgeon wanting personal acclaim. It can be the company trying to hit a stock measure. It can be the company wanting to protect its intellectual property by blocking others because it's demonstrating clinical progress and therefore you can't do a workaround and get their IP. So there's a whole bunch of reasons for it.
Yeah.
It's an expensive game.
But it does, if done properly, it does get better, as you mentioned. I think that's
Yes.
also an important point, is that by virtue of what you've learned and Jaron has learned, for example, you're now able to do things much more cost effectively than were
Yes.
possible 10 years ago.
Yeah.
I think that's an important point for the field.
And if I could just answer both of those last two questions with an example. That cancer program that we did, Keith, that was actually run by investigator-initiated earlier on. So it was very inexpensive, very long. It took 15 years to do the first, no, it was actually about 7 years to do the first clinical trial. Very long, just one phase 2. So it's a little longer, but then California Stem Cell came in, scooped it up. Now it's all compressed. No burn from the past, like the 200 million, etc. And so there are ways to do it.
Interesting. We have a question?
Can I ask one quick question from Paolo in Italy? I would like to ask Dr. Keirstead if you had analyzed the advantages and disadvantages of doing clinical trials on acute spinal cord injury versus chronic spinal cord injury from a clinical point of view and a financial point of view.
Yes, I've thought about it a great deal. I think the field's been focused on acute because there are more transient therapeutic targets. So you may be able to intervene with neuroprotection, with free radicals, with secondary degeneration, with general immune. These free radicals and immune responses go away after a little while, and then you're left with a great big hole. And so it's a totally different approach. So it depends really what you want to do. The early interventions with acute are generally aimed at lessening the severity of the damage by stopping the secondary cascade, whereas the more chronic approaches, which are really just beginning now, the attention on chronic spinal cord injury is really new. I got one of my NIH grant applications sent back, and the review was one line from one of the reviewers, and it said, "Why don't you focus on acute for now?" That was it. That's all he said. It was incredible. But now that review would never come back because people are starting to focus on chronic. But it's a risk to the patient population and potential of outcome that's difficult to assess in an acute setting because you really don't know if the patient was going to get a little bit better when they're injected at 6 days. It's also harder, the quality of life assessments, which is one of the outcome measures. So I'm in a chair, I just got injured, 6 days later I get a treatment that increases my function by 30%, but I still can't pee on my own, and I'm pissed off, and I don't really like you and your research and your clinical trial because you didn't get me out of a chair, and I didn't know what it was like not to have that other thing, so I'm still disappointed in the trial. So these types of things are really difficult in the acute setting for patient management. I can keep going on, but I don't know. It's a big, broad question, but a very good one.
Are there any others online?
Yeah, I have a lot, but I don't know if we need to limit it because of time. I'll ask one more. This is from Mexico. It's pretty obvious that the finance of the research has to change. Would it be possible to finance the R&D, as in recent cases of Alzheimer's, Lou Gehrig's, or some other low-scale recent developments?
Is it possible to finance the R&D? Yes. I think the question is alternatively, in alternative means. And one of the primary ways is donations. So I think that is an alternative means that
The
has...
March of Dimes approach?
Yeah, exactly. Donations can very, very, can have a huge, huge impact. Like the donations to my laboratory of $50,000 was responsible for that first one, the CXCL10 treatment. $50,000 from 4 different donors, and that turned into over 35 million spent, and it's in the hand of Bristol Myers Squibb, and it's now got a life of its own, and it's going into phase 3. But it was a few individuals putting up $5,000 and $10,000.
Any more, Ryan?
Yeah. this is, okay. Can you, what according to Dr. Keirstead are the main challenges to doing clinical trials in low-cost countries such as India, China, or Mexico?
The major challenges, one of the primary ones is doing it in a meaningful way that is respected outside of that country. So note that it said clinical trials in that country, not a clinical treatment. So I'm not denigrating the ability of those countries to conduct perfectly good treatments and have excellent doctors and nurses and regimes. One of the great challenges is the lack of regulatory oversight, so that when that bona fide individual in Mexico does an excellent trial, is it actually respected by big corporations outside of the country or within the country as equally as a clinical trial that's run and certified, stamped beautifully by the FDA. So that's a challenge because, again, you've got to fund it. So those groups are generally left with fewer funding sources, and one of the primary ones they use is by charging the patients. And then you get into a moral dilemma of charging people to be test cases. You know, in the United States you can't charge for phase 1.
So thank you so much.